Okay, I see a lot of people starting to log on. We're going to start in just a couple of minutes. We'll start at noon sharp, so thank you for coming. I have that it's 12:59. We'll start in about 1 minute. People from my lab joining, which is good, always a good thing. So for those of you just joining us, we will start on time. We'll start about noon Central Time. I'm just admitting some more people. Thank you for coming. Okay, well, I do have that it's 12 o'clock sharp. I'm going to pass the host back to you, Dr. Perry.
Okay.
And you should be the host.
Okay. And you can see my screen.
Yep, everything's good. So good morning, good afternoon, good evening, everyone. Welcome today to our Dementia Journal Club. We're broadcasting live from Winnipeg. I'm Benedict Albensi, your Journal Club host. And really, it's my pleasure today to introduce Dr. George Perry. So Dr. Perry has been with us here before in Winnipeg. I think it was 3 or 4 years ago. Dr. Perry is a professor of biology and chemistry, and also the SEMS Foundation Distinguished University Professor or Chair in Neurobiology and former Dean of Sciences at the University of Texas in San Antonio. Dr. Perry has studied Alzheimer's disease since 1982 and was really the first to discover that oxidative stress is a key feature and related to neurodegenerative diseases. His studies identified oxidative damage and its source from metabolic and mitochondrial failure and catalyzed by iron and copper. This work led to a novel interpretation of the role of amyloid. That is, instead of causing Alzheimer's disease, it's a protective antioxidant response and the reason all amyloid-based therapies have failed thus far. So he is distinguished as one of the top Alzheimer's disease researchers with over, I think, 1,500 publications now, if I have that right, and one of the top 100 most cited scientists in neuroscience and behavior, and also one of the top 25 scientists in free radical research. He's received over, or he's been cited over 97,000 times and recognized by ISI as a highly cited researcher with an H factor of 157. Dr. Perry is also founding editor of the Journal of Alzheimer's Disease, where he's still the editor-in-chief, which is actually the most widely cited Alzheimer's journal in the field. He also has a long list of awards, including the, I guess these are more recent, the Roos Whipple Award from the American Society of Investigative Pathology, the Distinguished Texas Scientist from the Texas Academy of Science, the Society of Advancement of Chicanos and Native American in the Sciences Distinguished Professional Mentor Award, and the list goes on. I mean, there's really several awards. And I would be remiss at not mentioning, he's also mentored over 100 students and fellows in research. So with that, I'd like to introduce Dr. George Perry. Thank you.
Thank you. Thank you, Ben, very much for this chance to speak here today and join you. Are you able to hear me?
I can hear you and see you fine. I think you're on track.
Okay, good. I just wasn't sure of that. I know when I offer classes, I'm always checking and feel it's redundant. Then I find out 5 minutes in, they're raising their hand. Where did you go? Any case, what I want to talk about today is about how there's been so much effort to try to find a cure for Alzheimer's disease and why it may be much more elusive than we thought in the past. Not unsolvable, but more elusive and more complicated. So I guess I'm the one who's letting people in. Anyway, in search of a cure for Alzheimer's disease. And that's really what's happened with the field. I came to the field in the early 1980s. And in the early 1980s. And I would say not too far after then, therapeutic trials began with various approaches of different agents. And by the mid to late 80s, there was a major focus on agents that removed pathology. Over that period till now, my estimate is there have been at least 130,000 high-profile papers, probably much more. That was an estimate of a few years ago. 60,000 investigators have published at least a few papers in the field. And there have been 112 years of research and a lot of focus, especially recently in the United States. There's around $3 billion a year being spent by the NIH, as well as numerous billions from industry, as well as foundations. Yet all of the current therapeutics are based on ideas that were well developed by the time I entered the field in the early 80s. That is the cholinergic hypothesis, as well as the excitatory idea. And those approaches, which led to Aricept and Dementia, really only address the symptoms of the patients. So they have some value, but they don't benefit patients for any length of time and don't change the course of the disease. And in more recent times, there's been an incredible failure rate over the last 15 plus years, 15 to 20 years, in which 99.6% have failure. That was an estimate of Jeff Cummings a few years ago also. It's probably 99.8% now because there have been more work done and no new drug has been approved in recent times. So leading me to think, is the focus stay too long in removing the pathology of the disease? And there's two ideas of that the plaques and tangles are really the disease itself, or they're completely irrelevant. And then I'm going to present a third view, that they're really important, but they may not be the causative agent of the disease. But the only way to really know this is what's been possible in the last couple of years through the advent of Biogen and Lilly that developed antibodies that actually were able to remove amyloid beta from the brain of living patients and be able to follow those patients cognitively, as well as look at amyloid levels by imaging techniques. And what was noted was that I wrote down here, no clinical benefit. It's being argued that there is a small clinical benefit, but the clinical benefit is not an improvement. It's a change in the rate of decay, and it isn't clear that it's clinically meaningful or that it's real. And that isn't really what the amyloid idea is driving the disease promised. They promised that patients would actually get better and move closer to normalcy. So where do we go with this? Do we just do better ways to remove amyloid? Do we try to get the disease earlier, which is what people have been advocating? I mean, I've gone to the point of saying, well, then you need to remove it from in utero, even maybe while people are dating before they even have children. When is the right time to remove amyloid? What form is to be removed? Or are we down the wrong target? And the same logic is being used for tau also. And this doesn't mean that the tau approach, which is the fundamental component of the neurofibrillary tangle and its removal, at least in some sphere, may make a difference. But is this just going to end out with a major focus for 20 or 30 years and get nowhere? So how we came about of thinking that the plaques and tangles were responses to the disease, and actually responses that are homeostatic, was in our studies of oxidative stress. And I'm going to simplify a lot of studies here, and with the mitochondrial studies, I'm going to really focus on just a few studies that sort of keep to the point of what's causing what we're noting here. Well, one of the things we were looking at was we thought initially that oxidative stress would involve plaques and tangles in the pathology of the disease because they have long residence times. And some indications from some biochemical studies that were not done by us and cell biology studies are that the plaques and tangles may have residence times like 20 years. So we thought they would build up a lot of oxidative abnormalities just for lack of turnover, much like blood vessels do. Well, that in part is true. But after a couple of years and developing many markers, we started to notice a second pattern, and that's what I want to talk mostly about here. That while tangles, these are changes within neurons that I'm mainly going to focus on at this point, that they do contain sugar-related modifications that are increased resistance for removal. But they also have other types of changes, such as protein direct oxidation, lipid peroxide adducts, as well as surrounding cytoplasm as 8-hydroxyguanosine. And when we looked at this, we noticed that the oxidative changes actually were not in the tau changes mostly. They were in the surrounding cytoplasm in neurons of people that had Alzheimer's disease. And in fact, a normal neuron really only exists in a normal person. And I would add even further, a young person under 40, in which we don't see any of the phenomena I'm going to talk about today. When people become older, when they develop Alzheimer's disease in particular, even though the cells look normal, they have prominent oxidative changes for all of these things. And this broad range of oxidative changes that I'm showing here would be most likely produced by metal catalyzed hydroxyl radical formation, but we'll talk about that more later. So to find 8-hydroxyguanosine or lipid peroxidation or protein oxidation requires that the oxidation occur within exactly that cell. And so this further cartoon is showing that if we wanted to know what was abnormal. When we had neurons that had tau changes, such as you can find in pre-tangles, tangles inside neurons, and extracellular, it wasn't surprising we would have something like, well, it was an observation and it was nice to know, but it wasn't surprising because these neurons clearly have problems with their cytoskeleton and other things. But we found that people, the normal appearing neurons, had oxidative changes that were actually quite prominent, and quantitative analysis actually showed they were reduced in neurons with tangles. So this led us to look what is abnormal in a neuron that looks normal at the light microscopic level, okay, superficially. Does it have changes in metals? Does it have changes in amyloid? Are there changes in mitochondria? And I end on mitochondria because the earlier work of John Blas, Wentbiel, and others had suggested that there were fundamental mitochondrial problems in Alzheimer's disease. That even in cells from the body, fibroblasts or blood cells, they found changes in mitochondria and how they're organized. So we wanted to look at this the way we had for the oxidative changes. One of the critical elements was that we didn't do bulk analysis. We found out how to localize the oxidative changes using antibodies or histochemical stains or molecular probes so that we could know exactly which compartment they were in. And I'll mention more about that for metals. This turns out to be critical because many of the changes are very compartment specific. Well, and when we did that, we wanted to know if there were changes in mitochondrial DNA, because mitochondria have their own DNA that codes for some of the key enzymes. It's 16,000 bases, a circular DNA, very similar to bacterial DNA, prokaryotic DNA. And when mitochondria have problems, sometimes they develop what's called a 5 kb deletion. And we made a probe specifically for this 5 kb deletion and for the wild type at the area that would be deleted. And we looked we're looking to see if there was a change in this. And we also did in parallel PCR experiments and enzyme histochemistry and enzyme assays for mitochondrial enzymes. And what we found was really striking, because when we look for mitochondrial DNA by in situ hybridization, if we look at somebody with Alzheimer's disease versus an age-matched control, you'll see the signal is much stronger. Quantitation shows it's about 3-fold more. If you look at protein, such as cytochrome oxidase, immunoreactivity, you'll see that it's also increased about 3 to 4-fold. It doesn't have exactly the same localization. It's more diffuse and extends out dendrites and proximal axons, but it's increased. And yet at the same time, we found that activity of cytochrome oxidase, either by enzyme histochemistry or enzyme assay, was slightly decreased. And this really was a paradox for us of how you could have more DNA and more protein, and yet less activity. So we looked at biopsy specimens to see if there were more mitochondria and how they were organized in incident cases of Alzheimer's disease. These were taken at biopsy while the patient was living, and so they were taken for diagnostic purposes and when that was considered appropriate to be done, like from the 60s to the early 80s. That was done UCLA and other locations. In fact, some of these specimens came from Bob Terry, from his specimens he analyzed during the 60s. And we classified the mitochondria. So we did a montage picture. It was with negatives, not the digital thing that would make it a lot easier now. We made a montage of neurons and looked at the mitochondria within them. And we found that some of them had broken cristae. And we also quantitated these. Now, it'll be clear why I quantitated. This is lipofuscin. This is the vacuole with lipofuscin on it. And we quantitated these, and we found, if anything, total mitochondria is reduced. That would be all of the mitochondria with broken pieces, these two together, it's down. The normal mitochondria like this, slightly less, well, it's actually significantly less. The broken cristae, there's no difference. And my interpretation of that is that, and these have been described and people focused on them, I don't think that they really are anything other than fixation artifacts, because biopsies are not perfectly fixed. So the important thing to note, there isn't an increase in mitochondria. There's a slight increase in lipofuscin. It wasn't statistically significant, these vacuoles, but it was an increase. And why did we quantitate them? It has to do with where the DNA signal was. The mitochondrial DNA, not surprisingly, was present in mitochondria, and I'm not showing that here. We published it. But it was also present in these vacuoles, and it's an electron micrograph, and these are 20 nanometer gold nanoparticles that are present within these vacuoles. You notice also that they're diffuse within the cytoplasm too. So mitochondrial DNA has been released. That was true even more so if I showed the picture for mitochondrial protein or some of the prosthetic groups, antibodies to the lipoic acid. That's a prosthetic group for the a couple of key enzymes of mitochondria. Okay, so this is really strange. Why do you have so much of this, I call it mitochondrial garbage, accumulating in cells? Accumulating in cells that otherwise look normal. So then this really led to us on our focus on looking at mitochondria. And so we wanted to look at 2 of 3 important phenomena. We've studied, so there's transport of mitochondria, which is the lower part of here. Mitochondria move from the cell body, which is denoted here by the nucleus, and they can move out to axons and dendrites, so they can return back, and that's antegrade versus retrograde. But they also have another phenomena that's more recently been a focus of many research, and that is they also can fuse together to form one structure, or they can pull apart and form 1,000. And that you can see here when you look at cells that have been, this is a particular cell, and if you have different proteins, and I'll mention some of the proteins, but not all. If you have them pull apart, you can have 1,000 mitochondria in a cell, or you can have one mitochondria in a cell, and it's a thread-like structure throughout and all connected. And this is another part of dynamics. The third part that I won't present today is you can also look at turnover, which is also altered, but I won't talk about today. So initially, you wanted to look at transport and look at this in a structural way, and then lead into more dynamic experiments. And what we did was to look at the same micrograph where we looked at the mitochondria, and we counted the number of microtubules that were present within the cell body. And the microtubules in the lower portion here are marked by the red arrows. And you notice this is a neurofibrillary tangle made of paired helical filaments. And in the same neuron, you can find microtubules. We measured the full length of microtubules or the number of cross sections, and both were analyzed, and both had the same pattern in case there was a change in shape. This is the nucleus, that's a mitochondrion. These are ribosomes, polyribosomes. And what we noted was an incredible decrease in the microtubule density in neurons from people with Alzheimer's disease. You can see here the control density, which is in blue, versus those that from people that had Alzheimer's disease, which is much lower. There's no statistical difference if that neuron contains a tangle or not. Okay, that's an important element. And if there are questions, I have some reasons why I think this is the case. We also have an internal control for fixation and other things because we have other cells, neurons that are not the pyramidal ones and are much less affected by the disease. And there was no statistical difference between microtubules and them. Also shown here, if you looked at this with aging, just among the controls, you can see a decrease. And that's been shown by others also. That, well, there were a couple studies that had been done looking at tubulin levels, and they also found this decrease. And I could mention in study, more recent study in the last few years, looking at tubulin levels, and also we found that they decrease. There's a change in microtubule stability. Having this data, okay, we thought the next thing we really needed to do was to model it so we could understand it in a cell or an animal model. So we embarked on using cells in which we lowered microtubule density to see if we could find the same type of microtubule changes. And we did not. We're not able to recapitulate this in a model, at least the ones that we used, which was nicotocell. But at the same time, we started looking at fusion fission. And we looked at the proteins of fusion fission. One is that they are also, I'm not showing it here, but they co-localize with the large neurons that show the mitochondrial change, so the dominant ones that have it. And if we show the staining, there's a decrease in Alzheimer's disease for some and an increase in others. I'm showing instead just the blot data. So if you look here, these are controls, and I'm going to focus on one protein pretty much. Dynamin-like protein 1, which plays an important role in controlling mitochondrial size, and just a nice one to follow through. And you notice between the controls, it's very prominent, and the Alzheimer's, it's much less so. In fact, it decreased about 75%. Not all the proteins decrease. FIS1, protein important in fission, actually goes up. So I just want to show it's not a global, and then there's actin for loading. What was really, and that is in brain tissue, but when we went to look at cells from patients that had Alzheimer's disease, that had Alzheimer's disease or didn't have Alzheimer's disease, these are aged individuals, and the cells are obtained from the NIH repository. We found that when you looked at how their mitochondria are distributed. And now these are living cells, so we're able to use different types of stains. We found that you could find mitochondria often to be collapsed around the nucleus, much like those damaged mitochondria were that I showed with the DNA, as opposed to dispersed. And if the cells came from somebody with Alzheimer's disease, there was about 15–20% of those collapsed, as where if they were from an older normal individual, it was about 1/3 of that. And not shown here is that we went and analyzed the same proteins, dynamin like 1 protein in Alzheimer versus control, and we found it reduced. Now, in more recent studies, which I haven't published and I'm just showing here, we took the fibroblasts and differentiated them and iPS cells and turned them into neurons. And we were also able to find the same result in neurons that are created in vitro from Alzheimer patients and see a difference. Okay, let me just wait. Okay, so we were able to find these same changes. And so why is this important? Well, and how does this link to other things? And that's what I'm going to say some of the pieces here and then more later. Well, mitochondrial dynamics is really critical for how cells deal with mitochondrial damage, because the mitochondria has less DNA capability of repairing DNA abnormalities, and there are multiple copies. So one of the ways it does that is to mix the mitochondria together to through complementation save them, or becoming smaller pieces so that they can be destroyed because they are defective and go to autophagy. And another part that's really critical is that the transport, if you have 1 mitochondria per cell, how are you going to get it into an axon or dendrite? It's going to be too large, even structurally. So dynamicism is really important. So we wanted to understand how this played out. Number one, we went back and looked at the biopsy specimens at the proximal axon. We found that there were big distances between mitochondria, and I'm not showing those pictures here. When we did this in vitro, that's what you find. You find big distances between mitochondria when you alter dynamin like 1 protein. And this can be directly tested as to what is the driver of this. Well, we know that amyloid precursor protein and its production of amyloid is an important element in it. So if you take cells and you express normal amyloid, overexpress it, you will end up more of those mitochondrial problems. That clustering, basically it's a clustering and larger mitochondria as they're more fused together. Another part is you can look at their actual dynamics, and that can be done by bleaching. So we took cells, we bleached them with a laser, and we watched the recovery. If they are overexpressing amyloid precursor protein, it takes them several-fold longer, at least 3-fold longer to recover for the bleaching. And further is that if you compare with amyloid that has a mutation that produces the amyloid precursor protein that has the mutation that produces a higher level of amyloid, it even is more so. It will create this. And it's blockable by inhibitors of amyloid formation, beta secretase inhibitors. So all of this piece, I've simplified to say that normally, especially when in a younger individual, the mitochondria are distributed throughout the cell wherever energy is needed. Probably a lot of them are near synapses and all of the other parts that maintaining the electrical potential of the neuron. And during aging process, and more strikingly during Alzheimer's disease, there's much more paranuclear clustering of mitochondria that are damaged. There are bigger distances. We've actually measured it. I'm not showing it here. In the same micrographs that I showed earlier, we had the proximal hillock. And so there's bigger distances. And we think this plays a role in changing the energetics, but also plays a role in mitochondria turnover. And what are mitochondria full of? As we'll come to later, they have a lot of their respiratory enzymes, and respiratory enzymes have copper and iron, and some cases manganese, but I'm going to focus on copper and iron. Okay, I want to switch to another topic and then I'll return back to that. So the mitochondrial changes occur in the same compartment as the oxidative changes. They're especially, the abnormalities are especially in large neurons. Now I'm going to show the first time I've shown primary data what an immunostain looks like for oxidation. And we looked at how does oxidation relate to the pathology. And I mentioned earlier, when we looked at it compared to tau stain, it's actually reduced by 50%. It's also reduced significantly by amyloid analytically. Okay, so if we look in this case, I'm showing Down syndrome. Down syndrome, people develop Alzheimer's disease-like changes, plaques and tangles, and the brain is essentially indistinguishable from somebody who has Alzheimer's disease by the age of 35 years old. And, but they start to show other abnormalities, little bits of amyloid, even in their teenage years. So we looked at a series of Down syndrome cases, thinking that was a good way to understand when oxidative changes occur, when mitochondria changes occur, and how they relate to amyloid. And when we did this, we were really perplexed because the cases that were older, and 61 is old for somebody with Alzheimer's, with Down syndrome, we couldn't even detect the neurons being stained by 8-hydroxyguanosine, which is RNA oxidation, mainly ribosomal RNA oxidation. And yet, in the teenagers, we could see it quite prominent. We quantitated this and found that those that were 0 to 9 years old had very low levels. It went up in the teenage years, even went a little bit more in the 20s. But when we analyzed people over 30 years old, it was less. Important to note, and we quantitated the oxidate, and we quantitated amyloid, you'll see that it actually only starts to really go up in the 20s and 30s, that transition period older. And then if you correlate just those cases with amyloid versus 8-hydroxyguanosine, you see a strong negative inverse relationship. And that's what I meant about, we were really perplexed about this observation. Why is it negative? And the way in which we quantitate the extent of staining is done on a per neuron basis. We don't just scan the whole thing. We actually scan each particular neuron. We exclude the nucleus. We go through the midpoint. So it's not about neuronal loss that causes this. Okay, so at the time we were doing the Down syndrome, we want to know, is this something we would see in normal Alzheimer cases or various types of people? So these are sporadic Alzheimer cases that were collected at Case Western, went to the Alzheimer's Center, and what we noticed was the same relationship. More amyloid present in the brain, less oxidative damage, only among those people with Alzheimer's disease. And the controls, it isn't there, and I can answer more about that later if there are questions. And just like it was in the Down syndrome, it's related to duration of disease. You start higher and you move down. Okay, there's a difference with ApoE, but both are significant for the drop. It's just that there's a slight difference because the people with ApoE tend to have more amyloid as the disease goes through. You see, these are more within a certain range, and this one goes to a much higher level. Okay, so it's inverse, not linear, but still significant. Didn't have it here, don't have a slide here, but we also looked at genetically caused Alzheimer's disease, found the same relationship, happened to be able to analyze some people that were pre-symptomatic and pre-pathology, had high levels of oxidation. So all saying that whenever you find amyloid, you find reduced oxidative damage. Okay? Doesn't fit the idea of it causing things. And then we tried jiving that with some other work that we were doing, particularly this study that Mark Smith had done, in which he developed a histochemical stain looking for redox active iron. I mean, and it's said to be redox active iron, but it's basically the ability to be able to change the redox state of iron. And we found that people with Alzheimer's disease, their brain is able to catalyze the oxidation or the reduction of iron. So it'll change iron 2 to iron 3, it'll change iron 3 to iron 2. And if you take a brain tissue from a control individual, strikingly if it's a younger individual, but an individual without pathology, you don't see any of this type of activity. So to address this further, we wanted to understand the mechanism for this. I mean, what was responsible for the redox activity? Was it iron-induced redox activity, which is what we thought it was initially? We still may be. It still needs to be defined more. So we wanted to understand where the iron was in plaques. And plaques are fairly easily isolated. This is some isolated plaques using a modification of a method developed by Dennis Salkow. You homogenize the tissue, run it through a few steps, and then put it on a sucrose gradient, and then you do a cell sorting technique. And the cores of plaques, when they're stained with Congo red, have this Maltese cross-like appearance when stained with Congo red and viewed in polarized light. And they stain with antibodies to amyloid beta, and this is the control for that. And then we did Raman spectroscopy because we wanted to know where the iron was. And when we did this, these plaques, we put them in the Raman spectrometer. It's a microscope that looks for Raman spectra. And we were not able to find any spectra of iron. What we were able to find was a clear peak for zinc, and for the small one for copper. This is able to be removed by chelator with EDTA. Not surprisingly, we saw a spectra of beta sheet. Okay, but what really perplexed us was that the iron wasn't there. And finding copper is also really important. The work of Ashley Bush suggests that copper played an important role in the disease, and we've done some experiments with him. And this is an experiment that we did independently with a group in Japan. And we looked at what happens when amyloid beta binds copper. The amyloid beta, at least in the synthetic peptide form, which is what's done here, is that it binds the histidine residues that are present in the amyloid beta. And when you take copper, will oxidize lipids. It'll act as a catalyst. It'll oxidize vitamin C, it'll oxidize anything that it has a reducing agent, and then is able to transfer the electrons. So if you add copper, and that's what this is, just pure copper, to arachidonic acid, a polyunsaturated fatty acid, you'll get lipid peroxidation marked by formation of aldehydes, hydroxynonenal, and at very low levels, and that's what the upper curve is. If you add amyloid beta to it at higher ratios, of 1 or less, it essentially blocks all of that oxidation. So this came with the idea that amyloid beta is playing a role in blocking that oxidation. Okay, well, where are these metals exactly located? And I'm going to show some of the micrographs, and we've done a lot recently and have a paper in a pending review right now with other techniques. And this was done in an atomic resolution microscope. And this section differs from what you're used to seeing for electron microscopy because although it's embedded, the plaque has been embedded in EPON. What is the big difference here is that it has not been treated with osmium, uranium, or lead. It's not been treated with any heavy metals, which means that the contrast that's there is the result of the endogenous contrast. And you'll notice here that there's little small dots that are electron dense. Okay, if you stain them, you wouldn't notice them because the stain would be more dense than them. And when we looked at those dots, we were able to see that they have a spacing pattern exactly what you, in comparison, spacing pattern that's exactly associated with magnetite. And and then we actually analyzed them to see if they had magnetic fields. Because we knew that the particles themselves, plaques and tangles, are attracted to magnets, but we couldn't find that they had a direct magnetic field. And this study actually resolved this issue, because what it is they have a property called superparamagnetism. That means they have the ability to be magnetic when they're in a magnetic field. And so you could think that's a trivial thing, because magnetite should always be magnetic, but it isn't, because the size of the particles is right at the borderline for this property. If they were any smaller, if they were larger, they'd have magnetic fields, like if they were larger than 15 nanometers. And at 20 or so, 10 to 20, it's kind of variable. Anyway, we directly demonstrated that they have this property of superparamagnetism. Okay, we also were able to look at amyloid plaques and look at how they're held together. And when we put in copper chelators, we found that they fell apart. I mean, Ashley Bush had shown something similar to this, not directly with plaques, but more about how the solubility of amyloid beta in the pellet changed. But it falls apart. Let's see. And if you take synthetic peptides of amyloid beta and you add copper to it with some buffers, you'll end up getting things that look actually much like black cores. Now, that's right. Okay, go back before here. So we've done some studies, and I'd rather not. They're in collaboration with Joanna Collingwood in the United Kingdom, using the X-ray microscope, either at Berkeley or at Oxford. And in those studies, we were able to analyze the redox state of the metals in specimens much like this. And we were able to look to figure out what type of redox state. And we were able to find, and I'm now presenting our work also, that we found that the copper was associated with the filaments and the iron mostly with these dots, and that that the iron is, not surprisingly, because magnetite, iron 2 and iron 3. In the case of the copper, it is copper 1 and copper 2, which was really a bear to nail down, was able to be nailed by these studies. And both of those redox pairs are essential in order to be able to, for the metals to have the potential to cause oxidative damage, or alternatively, to also play a role in reducing oxidative stress. And that is actually what we think is happening. Both amyloid and tau, I could present the work there. It involves a specific enzyme called heme oxygenase, which is co-expressed with tau. But in the case of amyloid beta, we think that it plays a role both in sequestering copper and prohibiting redox cycling, or promoting redox cycling, which stabilizes iron, much like it's in ferritin. And those experiments are ongoing right now. Okay, so I could present a lot of different talks about things just that we did from our laboratory dealing with copper and iron abnormalities. You could talk about glucose transport abnormalities, reduce microtubules, I touched on these, increased inflammation, vascular degeneration, cell cycle entry, multiple signal transduction alteration, many related to signaling of oxidative stress like hemoxygenase, HIF1. But there's also, as I said, increased antioxidant responses. It isn't just that there's oxidative damage in those neurons. The neurons respond with every type of oxidative damage response. And probably the most fundamental of these is the induction of the pentose phosphate pathway. Okay, tau deposition, amyloid. So yet it's important to remember with all these really disastrous changes, which you think would kill everything, and we're definitely doing studies to further understand cell death and apoptosis and the newer process, pheroptosis, but the neurons still live, decades like this. And this leads us to think that we could go on for another 20 years documenting abnormalities that mark a change from having normal brain to having Alzheimer's disease. But are these numerous abnormalities instead marking something that's really pleiotropic? And is it similar to hibernation? And it's not in a sense that the brain in Alzheimer's disease hibernates exactly, but that there's a fundamental change in metabolism and that everything changes and that new change is stable. And that targeting any one of the abnormalities could imbalance neuronal survival. You really have to understand how the things all fit together. And then when you look at how many targets people have, and some of those relate to the things I've already mentioned, there's amyloid beta type change, there's, tau pathway changes, all sorts of things. I could say, oh, our work says that you should remove metals. It's more complicated than that. If you look at globally, do metals really change? The answer is very little. It's only in certain compartments. And many of these changes, even with the things like oxidative damage, may play regulatory roles that we don't understand yet. Okay. So I think that that's why we haven't progressed, because the things that have been most of benefit for patients so far have been things that restore metabolism, exercise, stress reduction. All of those are saying about a better homeostatic balance. I wouldn't want to leave you with the idea that that's the only thing you need to do is to do meditation. What we really need to do is understand how all those things fit together and not try to unbalance everything. Okay, so there's, all of this stuff we're doing, all this work, and of course, those of you involved with companies know that this is a valley of death, academia's ideas, great ideas, like all these ones before, and then you try to convert them into a drug. And they don't always work. So I don't want to leave you with the point that, that there's not going to be a promising drug for Alzheimer's disease. I think there will be, but I think it's going to be much more like what has happened for heart disease, where it's a combination, where lifestyle plays an important role, but not a total role, and drugs play an important role, and surgery plays a role. I don't know, and maybe that's deep stimulation for Alzheimer's disease. And that you may have to put all of these together in order to make a tremendous change for people. Because all of us want to have the magic bullet, but there may not be a magic bullet of a single thing. So oxidative damage is an early feature of Alzheimer's disease. I presented some data for that. I left out some of it, but we know that the highest level is during MCI for sporadic Alzheimer's disease. Mitochondrial abnormalities are critical to oxidative abnormalities. They actually correlate with them both topographically and quantitatively. Amyloid and amyloid precursor protein are linked to mitochondrial turnover. And even if it's not causing the disease, it's a critical part of the pathway of moving forward. And copper and iron abnormalities are intimately associated with the pathology. Both plaques and tangles are really wrapped up in these type changes. And finally, a cure, whatever that means, it basically is a reversion to a normal path of aging, which people are functional, requires understanding and targeting the complex compensatory abnormalities of Alzheimer's disease, not removing them. And thank you very much for the attention and for the invitation, Ben.
Okay, thank you, George. I already start seeing some questions showing up in the chat box. I just want to ask a question of my own real quick. You showed a lot of your own data, and I was just wondering if you can make any comments about any potential sex-based differences in some of the data that you've shown today in these slides, or was it worthy of?
No. We didn't publish it. We actually had that data. I don't know why we didn't publish it. But, yeah, I'm trying to remember exactly. Women have more amyloid, so the curves are slightly different when you end out segregating it to females. Beyond that, I mean, we did a lot of work on gonadotropins but that was done not so much for oxidative abnormalities. The other part, we segregated that. We didn't segregate people as to race or ethnicity because we didn't have enough people that really were fit into different categories. We did in the gender.
Okay.
I think they're kind of evenly divided between males and females from the brain bank. I just don't remember though.
Okay. So yeah, I've got a few questions in the chat box. I'll repeat some of these, and then also we might have time for maybe 5 minutes of questions or so. And so you can unmute yourself if you want, or put your question in the chat box. And I'll just remind everyone that we did record today's presentation, and I'll make that available online through social media later using LinkedIn, Twitter, and Facebook. And so if you follow me, you'll see those links. So the question from Dr. Musavi here, it goes, so do you think thin oxygen therapy could be, effective to reduce oxidative damage in early Alzheimer's?
Not a straightforward answer, because hypoxia, low oxygen, can cause oxidative stress, and high oxygen can cause oxidative stress. I've heard of people telling me anecdotally that people going into hyperbaric chambers had some benefit, but it's very anecdotal. So I really don't know the answer. Oxygen therapy would, if anything, likely increase oxidative damage, but it might improve respiration to some degree. That's the reason I don't know if it wouldn't be a benefit to patients. But I don't know of any really well-controlled study on this.
Okay, so other questions, either by chat or if you want to unmute.
Hi, Dr. Perry. This is Sanjeev. So my question to you, like at the very beginning of your talk, you said like amyloid beta has been removed from the brain with no clinical benefit, right? So what's your take on like aggregated tau or soluble or insoluble tau? Do you think like they're protective, or if we remove them, they can be beneficial?
You know, when Mark Smith and I suggested about the year 2000 that this approach of removing things wouldn't have benefit, we were shooting from the hip a little bit, okay? You know, not aiming completely, is that we thought there was a good probability that these were compensatory proteins. I think that both proteins are acting much like inflammation is acting. So it depends on context. So even amyloid modulating the amount or in the compartment could have some small clinical benefit, but it's not going to be earth-shaking. And I think it'll be the same for tau. These are normal proteins that have key functions. In the case of tau, has a ton of functions. So I kind of have what I've gotten out of more studies of these, the proteins that we focus on in the Alzheimer's disease or prion disease, or any of the rest of them, those may be the critical proteins that are essential for normal aging. the reason they're, as we get older, and older meaning probably beyond 40, something like that, sort of post-reproductive period, that our brain has to do a ton of compensations to remain normal. Because all of us know people that are 80 or 90 years old that are cognitively normal, in addition to demented. And isn't it amazing? If we looked at their brain wouldn't look like a 20-year-old. So that's what I view, that these proteins are probably the key ones. The other thing, the insight I had, which I wish I would have gathered 20 years earlier, is that genetics doesn't tell you causality. People push for it, saying that it causes, and they always say that, but it doesn't show that. It shows association. It's a statistical association. The causality is something you have to establish. And the mice do not establish it, because you're creating a problem. That's the thing. You put the mutation in a mouse, and the mouse has some disease. It's something like Alzheimer's disease, if you imagine strong enough. Okay, but those are not normal proteins for that mouse. It screws them up. And then if you take them out, they get better. But for us to have amyloid and tau in our brain when we get old is a normal feature. So could there be a therapeutic that's related to them that has benefit? I think so. But that therapeutic might even increase the level of the protein. Not saying that would be what I would advise people to do, but you just don't know what it's doing, or it could change the compartment for its organization. The same issue, what we think now, and we don't have good evidence for it, but I think we have some, certainly we have chemical pieces, is that we think that the copper that's released from mitochondria, instead of just being turned over, because copper usually is sequestered very quickly into other proteins, chaperones, so it doesn't cause oxidative damage. But maybe it's sequestered into amyloid beta to play a role in redox stabilizing and that this is part of a normal process, except you don't see it. When mitochondria are turning over all the time, copper is being released all the time.
Hello.
And how do you go about stabilizing it?
Okay, thank you. So I think we have time maybe for one or two more questions. Anyone else today?
Hi, my name's Andra. I'm a grad student in Dr. Carl Cotman's lab. I was wondering what you thought about people who don't have amyloid plaques when they get older. Do you think that those people are more resilient to oxidative stress if your hypothesis is correct?
Well, what's interesting, and part of it's in my graphs, and I've had that data for a long time and not published it. We also did a study looking at what type of changes occur in people that don't have, that don't have Alzheimer's disease and have plaques. And one of the things is that plaques always, they correlate with the amount of amyloid deposition you see directly, with an R squared of close to 0.9, which is not true for Alzheimer's cases. I kind of led one to believe that, but that isn't true for Alzheimer's. For those people that lack amyloid, you mean like that are 80 years old and have no plaques?
Yeah.
I don't know how many there are of those, okay? Because most people have some. I don't know. I think there are a lot of different paths for aging. I know when we've looked at population-based studies, analyzing the brains from people from the Rush aging study, you found a lot more variation in patterns than you do when you analyze people that come to a university hospital, which are much more segregated. So I don't think I've answered your question because I don't know the answer completely. I haven't studied it.
Thank you. Thank you very much for considering it.
Hello, Carl.
Hello, Barry, George.
Great to see you.
Yeah,
Since I don't see anybody anymore.
Good lecture.
Oh, thank you.
Yes, so I think we're about to wind it up. Thank you again, George, for your time and a great presentation. Always great to see you. Oh, we do have another question.
Yes.
So let's squeeze this in if that's okay.
Yeah,
Okay.
no, I'm fine.
This is from Victor.
Yeah.
Hello,
Well, thank you
Victor.
very much, Dr. George Perry, for the interesting talk. I was wondering, what do you think about the MCI subjects in the progression maybe that they are having into dementia? Are these effects also occurring?
Yes. Okay. So we obtained cases from, we actually initially wanted to study it as totally incident cases in a population-based study, and I did it with the Rush brains. The data was really not so easy to understand, so it still hasn't been published. Then I embarked on incident cases that were from Washington University. And they have the highest levels of oxidative damage during MCI. In fact, pre-MCI is basically the same as normal.
Yep.
And MCI goes up and then it goes down. So every result is the same. It goes up and then comes down. But something happens during MCI from the data, and that's the paper I haven't published. I've had it like the longest residence time of a manuscript written that I have not even submitted ever. And that is that the relationship between oxidative stress and amyloid is different for normal people than for people with Alzheimer's disease. And it changes during MCI. What I think happens during MCI or during the onset of the disease is that there's a fundamental change in metabolism and that everything changes. And you're seeing a pleiotropic shift, and you have one relationship. And if I had to guess, that's what I think that's where the induction of things like changing glucose metabolism from producing ATP to reducing the equivalents for antioxidant defenses. That's what the pentose phosphate pathway does. But it's probably much, much more. All of these changes we've documented over the years pop up during that period, and they mark that everything is different. And you know, if we had quantitated other things, we probably would have seen that they change also in these. So you don't really notice it unless you quantitate things. And the correlations are real. And during normal aging, amyloid and oxidative stress, which are both very low, because we find amyloid after age 40, and you can quantitate it, and you can find oxidative stress after age 40, that small amount is highly correlated with each other and positively. So I think that what happens is that when you're normally your age, you're producing low levels of amyloid and it's doing its job and it doesn't cause a problem. It's not associated with plaques, and everything just kind of works. And then suddenly at some point, the system decompensates and it really hyper does. And that would suggest that modulating amyloid, or I think much better, modulating the more fundamental problems, which are probably metabolic in nature. That's what I think the type of studies that Carl's done, many people are doing, really points to a fundamental metabolic abnormality that's much more subtle and is a part of why some people continue on to 100, like Carl's probably going to do. And some people are going to fail at 65. And then, of course, the genes and other things play a role of how you're calibrated at these different points. So I know I haven't completely answered your question because I don't know the answer, but I do think MCI, that's the basic thing, is a transition from being normal function to having an abnormal function. And Mark and I were very interested in the idea of this as being like hibernation, in which the metabolism churns to keeping the brain alive for a period of time. So we studied Arctic ground squirrels and tau changes and things, but Arctic ground squirrels do a much better job than us. They recover. We don't recover from Alzheimer's disease. And that's what we need to do. We need to figure out how to turn things back And I do think from studies that Carl and others have done, if we were able to reduce the problem pushing things this way, people should actually have some degree of recovery.
Yeah, it makes sense. Definitely.
Right? They shouldn't continue to decline. They may, I mean, people recover from stroke, they recover from many brain damages.
Okay, so I think we're about out of time. We're after the hour. It's 1:05. So thank you again, George, for a great
Okay.
presentation. Always great to see you. Thank you so much for your time
Pleasure.
today. And thank you to everyone joining. Like I said earlier, there is a recording today, and I'll make that available shortly later this afternoon on LinkedIn, Twitter, and Facebook. So stay safe. Thank you. Bye-bye.
Okay.
You.
Thank you.
Bye-bye.
Bye. Do I need to do anything with this for saving it or anything, or just sign off? Okay. Just wait, let me see.